Okay, we are going to do some of the biotechnology chapter 8. Big chapter. I'm going to go into more detail in class. Okay, definitions. Basically, it's modifying genomes of organisms. It occurs in nature. It occurs in a laboratory. Please do not believe that this is just man's manipulation. We do it on a much more systematic scale than in nature, but it's been happening far longer in nature than we've been doing it. The main reasons that we do it are under purposes: production of vaccines and production of proteins. The two main things we're interested in. For example, all of you in healthcare are going to be getting the hepatitis B vaccine, or you've already got it. 3 vaccines. That vaccine is totally made by genetic engineering. More and more, that is going to be the case. Many therapeutic proteins made by genetic engineering. All insulin is is prepared by genetic engineering using E. coli. All right, so let's get into it. Here is kind of the big picture. We're going to get some drawing tools here. We're going to take donor genetic information right here. Let's say they're human chromosomes. And somewhere on here is a gene for insulin. And let's say that gene is for insulin is right there in black. We're going to cut up the DNA into fragments. And you see that fragment right there has the gene we're interested in, but we don't know that because you can't tell one fragment from another. We're going to take these fragments and insert them into plasmids. Now, we've talked about plasmids before. Lots of bacteria naturally have plasmids. Remember that they are not essential to the cell for the cell to live. They are extra potential genetic information. They can be lost, they can be gained. Well, you can get them out of bacterial cells and insert genetic information that you want to be in them, making a new recombinant plasmid. We've inserted a fragment that we hope, in this case it does, have an insulin gene on it, but it could have been a different fragment. It could have been that fragment right there, which had different genes on it, not production of insulin. But in this case, we've made a recombinant plasmid. We're going to put it now into a cell, a new cell, like E. coli. And we are going to get the E. coli to read the genes on that plasmid. Now, what we're aiming for is production of proteins like insulin. It might be that we just want it to replicate the genetic information for other reasons. But we're mainly going to go this scenario over here on the right. Okay, so what do you need? Let's talk about where you get this DNA. You can, actually I should include one extra thing here. I've got source of DNA on here. You could use synthetic DNA, and you could use mRNA to make DNA. In other words, working backwards from mRNA, you could make cDNA, complementary DNA. Or you can get the DNA from cells. I don't have that listed there, but that's rather obvious. In fact, that's the main place for it, is getting it from cells. Then we need to cut the DNA into fragments. I've mentioned that. And we're going to use enzymes called restriction enzymes, or sometimes called restriction endonucleases. They split DNA molecules. So look at the picture over here on the right, and you will notice a double strand of DNA right there. And you see the sequences on both strands. We are going to use an enzyme called EcoR1. These enzymes have peculiar sounding names, maybe. This one comes from E. coli. The first letter E for Escherichia, CO for coli, species name. R has to do with the strain that it was first identified from, and 1 has to do with whether it was the first, second, or third, or whatever enzyme that was isolated. It's called EcoR1. There are other kinds. There's BAM and Hindi and a whole lot of other ones by the dozens. Interestingly enough, these enzymes made by bacteria that split up DNA are a defensive measure of the bacteria that the bacteria have against invading DNA. For example, against viruses. That's what viruses do. They insert their own genetic information to take over host cell machinery. Anyway, so on this example, notice a section right here. The sequence from left to right on the top is ch8, and the sequence on the bottom strand from right to left is ch8, same sequence left to right, right to left on the opposite strand. The enzyme will cut between guanine and adenine on both strands, and you can see where the cut mark is there. So there's the cut mark between these adjacent A and G bases. The hydrogen bonds that hold the two strands together in that area break apart, and you end up with a split molecule right there. Now notice these little unbound areas right here and unbound areas right here. These are called sticky ends. Now if you stick them back together, they will reanneal and reattach. Or you can insert a new piece of DNA which has the same sticky ends, the same exposed bases, and the new piece of DNA will be taken up in this other DNA piece. That's the idea here. Okay. So here's exactly what I just took you through right over here. The section that we are putting the new DNA in is this greenish one. This is the piece we've taken out. We're going to insert it right there. In other words, we're going to make molecules that are recombinants. These base sequences that read the same way left to right and right to left are called palindromes. So as you can see, here's our linear DNA, and it's been cut. There you can see our sticky ends right here. We're also going to cut a bacterioplasmid with the exact same endonuclease, and here's our cut piece of plasmid with its sticky ends. And since you have the same sticky ends here as here, if you add these together, you will get a recombinant DNA. Same sticky end pieces. It reanneals. And then you just need an enzyme, like a ligase, to come along. Actually you have to add it. And that ligase now reattaches at the free ends of the DNA, and we now have a recombinant plasmid. So we're using the plasmid as a vector to carry the DNA that we are interested in studying, or the fragment of the DNA that we think has the gene we're interested in, like insulin. So remember that the rest of the genes on the blue plasmid are bacterial genes from E. coli. They're not virulence genes, they're just genes on the plasmid that are really unimportant to us. It's the genes in the yellow section from the human DNA that we're really interested in. Okay, another thing that, so let's go on with the same thing. Remember that when we cut that DNA into pieces, let me go back here, up here at the top on our donor genes, we really, there's 46 chromosomes, and when we cut the DNA into fragments, we're going to get a lot of fragments, and most of the fragments don't have the genes we're interested in. We don't know where their genes are. So we may have to do some experimentation using a lot of fragments to find the fragment that we're interested in. So that takes us to a gene library. So if you look over here on the left, I've taken you pretty much through all this. Here are our recombinant vector plasmids, and notice that each plasmid has a different little fragment of DNA. We can then take these plasmids and put them into bacteria, for example, like transformation. We talked about that in an earlier chapter. Each bacterial cell now has a different plasmid. Maybe one of these has a plasmid with the insulin gene we're interested in. But we can test each one of the cells. We can grow cultures from each of the cells. Remember, the daughter cells will inherit that plasmid. So we could get a colony, for example, growing from this guy right here. And let's say the insulin gene is right here on that little, black piece. All of the daughter cells will make insulin. So we could identify it very easily if that's where our insulin gene is located. That's called a gene library. Okay, so let's move to another really important tool called PCR. You've heard this before. It's been used in a lot of trials, and it's used to amplify DNA to make more of what you've already got. But real-time PCR is used in identification. It's a combination of PCR and a DNA probe. So let's say you want to amplify the DNA. You've got a very tiny, tiny amount of DNA, and you need more of it to do testing on. You're not going to change the DNA. You're just going to make more of the DNA that you've got. That's called amplification. PCR stands for polymerase chain reaction. Polymerase refers to DNA polymerase. This is the enzyme that's used to make DNA. It elongates chains of DNA during replication. However, we're not going to just use any polymerase. We're not going to even use a human polymerase. We are going to use an enzyme, a DNA polymerase, from a thermophile. And that thermophile is Thermus aquaticus. It's been isolated. It was isolated years and years and years ago. This is a thermophilic bacterium. It was first identified from a hot spring in Yellowstone National Park. So we're talking about an organism that lives at near boiling temperature, and it has enzymes that are stable at almost boiling temperature. Now, this process requires heat. In fact, you can see the heat. Look at these temperatures, 94, 72, 78. You can't use regular human enzymes for this. Our enzymes would denature at these temperatures, so we are using thermophilic bacterial enzymes for the process. Okay, so basically here is our little bit of DNA, double-stranded, one light green, one dark green, that we start with. We want to amplify this DNA. So we start with a little primer to get things going. It's a little, actually, small piece of RNA primer. We are first going to heat the DNA to about 94 degrees Celsius, and we're going to open up the hydrogen bonds, break the bonds of the double-stranded DNA, so we now have separate strands. Here's strand 1, here's strand 2. The two strands that were double bonded above. We add our primer. We now add, by the way, this is all done in a machine called a thermocycler. And there are tiny little wells you can see inside here, like itty-bitty little test tubes. You add nucleotides like guanine, cytosine, blah, 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 blah. You add your DNA. You add DNA polymerase. And this little machine is computerized. You set it for how many cycles you want to do it. Okay, so now we cool it down to about 54 Celsius, and DNA replication occurs. The DNA polymerase that we are using, the Taq enzyme, standing for Thermus aquaticus, is this little pink guy right there. Okay, so the end of this replication of DNA, we've now made, on this bottom strand, we've made a light green strand of DNA. And on the light green strand of DNA, up at the top, which is what we had before, this guy right here, we make complementary strand right there. So we now have two duplicate copies of DNA right here. Two duplicate double-stranded DNAs. We've doubled the DNA. That's called one cycle. All right, we're going to do it again. We're going to heat to 94. We're going to separate the two DNA strands, this one and this one. We're going to add our primer. We're going to add DNA polymerase. We're going to add nucleotides. Remember, this is happening inside of a machine. And at the end of that, we're going to get 4. We have now 4 sets of the exact same DNA. We do this again. We do 8. Now, this machine is computerized, and it doubles, 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 doubles. So in just a few hours, you can get a massive amount of DNA from cycle after cycle after cycle. Okay, I mentioned that bacterial identification can be done with a PCR. Now, the PCR process itself just amplifies DNA. But after you've got all this amplified DNA, let's say we use a DNA probe, and we're going to look for a particular sequence of DNA that is unique for a bacterium we're looking for. Maybe we're looking for Mycobacterium tuberculosis. Maybe we think this patient has TB. So we got him to cough up a sputum sample. We did PCR on the sample using bacterial RNA from the sample. We have now amplified it many times, so we have enough of it to work with. We now use a DNA probe. A DNA probe is a small sequence of complementary DNA. Let's say that, let me draw a sequence here. Here is double-stranded Mycobacterium tuberculosis, and let's say that there is a unique section of the DNA of Mycobacterium tuberculosis, which is A, I'm just making this up, A, and of course on the other side it's T, and let's say that this is unique for this particular organism, and no other organism has this many repetitions of AT base pairs. We make a probe of this. It's going to be a single-stranded probe. Okay, let's get rid of that right there. So we have a single-stranded probe. We add it to the amplified DNA. By the way, we're going to heat the DNA up from our PCR to break the hydrogen bonds. So we have just pieces of single-strand DNA. We add this probe right here. The probe will attach to any sequence that it sees that is T. I should also say that this probe has a little, maybe a radioactive tag on it, or some sort of fluorescent tag that turns a color when it attaches, and that will allow us to visualize, see, identify the sequence of the DNA that we're looking for on the Mycobacterium tuberculosis. That's a DNA probe. It's not very long. It's only less than a couple of dozen bases long, but it's unique for the particular organism we're looking for. Okay. Let's see. I think I'll do some of this in lecture. All right. So, how do you separate DNA fragments after you've got them? Remember we cut, at the beginning I took you through cutting DNA into fragments. Let me go back over there, remind you. Okay, so up here at the top, donor genes, here we got 46 chromosomes. We got to cut them into different fragments, and then we got to separate them. Okay, so that takes us to electrophoresis. All right, so we have in this test tube of blue stuff right here, we've got 46 chromosomes broken up into fragments, and it's very unwieldy. So what we're going to do is we're going to place these fragments in wells in a gel. This gel is analogous for the architecture, kind of like a agar plate, except it's not for growing bacteria or anything. It's just simply an agar gel. The fragments of the DNA will move through the gel. And that's because there's a positive electrode down here, and DNA's got a negative charge, and it moves towards that other electrode when we hook this up with an electrical current. So these are where our samples are added right there. The fragments will move through the gel at different rates. Small fragments move fast. Big fragments move slow. Here is our finished gel right here. This is where we loaded them. Big fragments here, medium fragments here, small fragments there. We can go in and actually cut these fragments out and use them. We can put them into plasmids. So again, this is a way of organizing fragments. That's all it is. Electrophoresis, when you combine it with something else, it's used for identification, but its main use is to separate and organize DNA fragments. That's what you're looking at right here. Another way to look at the electrophoresis. And you see an example down at the bottom left. The fragments have been dyed with ethidium bromide. They are orange. They were loaded down at the bottom in the little cut wells, and the migration of the DNA is occurring up the gel towards the top. Small fragments up at the top. Okay. Another technique used a lot is called the southern blot. It's used for identification, finding a DNA, a sequence of particular interest, sometimes called genetic fingerprinting. It's used for identifying different microorganisms. That's one use of it. Particularly important when you're thinking that this is a microorganism that cannot be easily grown in a laboratory. For example, you wouldn't generally do this on bacteria because they can be grown easily and identified easily in a lab for the most part. But things like viruses, protozoa, worms, well, maybe not worms. They're easy to see in a microscope. They're eggs anyway. But viruses and some very picky bacteria, they use southern blot on. So here's the procedure. Okay, so here are our fragments of DNA we've cut cut apart with endonuclease restriction enzymes. We put them into a gel to separate them. There you go, right there. Now, the gel is very unwieldy. It would be like picking up Jell-O in your hands and trying to do something with it. You know, the Jell-O would fall apart, and of course the DNA would fall apart with the the gel. So, in order to stabilize your DNA fragments, you transfer the DNA fragments to a filter. There is a filter underneath the gel. Okay, so here's the gel right there. We put the gel on top of a filter right there. Then we put things on top of it to, I don't want to say mush, but I'm going to flatten out the gel against the filter paper. And in that process, the DNA fragments from the gel move to the filter paper. They get transferred because the filter paper is right underneath. It's almost like stepping with your shoe on mud, and there is a transfer of the pattern of your shoe sole onto the mud. It's very similar, actually. Now, here's your paper filter with your DNA fragments on it. We then use a DNA probe looking for a particular organism that we think it might be, like tuberculosis. Remember, the probe will stick to the unique area of the DNA. We use some sort of maybe radioactive probe or some sort of fluorescein dye tagged probe, and we can find which fragment has the gene on it, or if there is no fragment that has the gene on it, that tells us that it's the wrong organism that we thought it was. That's called the southern blot. And here's an example of a southern blot. Okay, so you've got your fragments of DNA. Let's say insulin gene. Now that you've got the insulin gene, what do you do with it? Well, you can take the fragments of DNA and put them into cells as fragments. Or you can take the DNA fragment and put it into a plasmid and insert the plasmid into a recipient cell via transformation. Or if you just have the fragment itself, you don't want to put it into a plasmid, you can put it into a host cell, recipient cell by transformation. Plasmids are more commonly used though because they're much more stable. Or another thing you can use is a virus. You can manipulate a virus, take its DNA, manipulate its DNA, insert the gene you want, repackage it in a viral coat. Remember, viruses take over host cells. You are now using a manipulated engineered virus to infect a host cell. While the virus is doing that, it's inserting the DNA that you wanted to insert. You are using the virus as a tool to get the DNA into the recipient cell. So, vectors are generally either plasmids or they are viruses. And what do you put them into? If you're making insulin, you're going to put the genetic information into bacteria. E. coli, for example, are used commonly. Now, hepatitis B vaccine, on the other hand, which is made of a protein of the hepatitis B virus coat, it's made in yeast cells. They use yeast cells as the recipient cells to make the protein. But you can also use animal cells, plant cells, things like that. So some of the things that are made by genetic engineering, vaccines, hepatitis B, good example. Genetic screening can be done with some of the procedures we've talked about, DNA fingerprinting using southern blot, and potentially gene therapy. They use viruses for that to carry the new DNA in. That's to correct, I'll say, defective DNA that people might have, like a DNA that carries genes that carry multiple sclerosis or cystic fibrosis or something like that. That hasn't been all that successful, but still being looked at in research trials. And you see a bunch of proteins here that have been made by genetic engineering. All insulin, all human growth hormone is made this way. EPO, erythropoietin to tell your bone marrow to make new blood cells, all made by genetic engineering, not made synthetically anymore, not gotten from animal cells anymore. And here's just a look at how you can use a virus, package a virus, to carry genes into host cells that you wanted to carry. And you can stay on this page longer if you like, but this is the end.